at www.global-once.org. Without further ado, let's extend a warm welcome to Professor Ding He, Professor Jiying Li, and Professor Penghui Li. We have two Lis. I'll make sure we know who you are. Okay, Ding, the floor now is yours. You have 30 minutes for your presentation.
Okay, thank you, Professor Zhang. It's my great honor to be one of the guests to, present some of our work. Okay, so actually, I'm so lucky to present this, work after Professor Zhang Yongyu, because he did a wonderful talk, of the big picture. But today, it's kind of in a very molecular level scale, micro scale. We want to explore, the fate of the marine DOM cycling from the molecular scale. So before my presentation, I really want to acknowledge some of these co-authors from my group and some, collaborators from the, international institutes. And we also want to acknowledge, the funding support from the NSFC and UGC from Hong Kong, and the Center for Ocean Research in Hong Kong and Macau, and the AOE. Okay, so hold on. So we all know that the ocean DOM is a vast reservoir of the reduced organic carbon. And if you check the pool size, it's comparable to the atmospheric CO2, right? So it's a huge reservoir, which justifies its importance in the global climate change. But another thing is not only because of its size, but also because of its, characteristics. So if we zoom in, we gradually know the marine dissolved organic matter is a really complex mixture of multiple molecules with different reactivity, right? So this, the left panel shows you there's something we do know, but there's something we know we don't know. Okay, it's a super complex molecular space. Within this complex, reservoir, a major fraction of dissolved organic matter called attention, which is called the recalcitrant dissolved organic matter, because it shows a carbon sequestration potential because of its quality and quantity. So the quality of this subfraction is one of the key characteristics. It's really old, okay? So the 14C dating shows the age of those carbon can be averaged as about 5,000 years. So when we're talking about carbon sequestration, we really want to make sure those kind of carbon can be sequestered for a long time. So if we don't talk about the time dimension, it's not gonna be that meaningful, right? So that's the major characteristics for this recalcitrant dissolved organic carbon pool. And in terms of the quantity, it represents more than 90% of the dissolved organic carbon, so which justifies a huge pool size. So there's lots of mysteries regarding with the RDOM in terms of its source, its transformation, and its fate. So when I'm saying its transformation, so we kind of wondering how the DOM is transformed from the labile source to a recalcitrant form, okay? And when we were talking about the fate, we kind of wondering if with the global climate change, how this kind of pool gonna evolve, right? So all those questions actually are linked with the basic underlying question. So can we find some biomarker indicators to represent this kind of recalcitrant DOM pool? So as we all know, the organic carbon in the ocean is really complex, right? So they include both living things and non-living things. For the living things, from the macro scale, right, you can see all the fish, the zooplankton, phytoplankton, bacteria, and archaea, which Professor Zhang liked most, right? So those are the living things. We can see they are made of the organic substances. But for the non-living things, if we zoom into the molecular level, there are lots of different molecules. Some of them you may be familiar with, the glucose, amino acids, and some complex molecule, including the DNA and RNA, etc. And another really important complex molecules I'm going to jump into is the CRAM, the carboxyl-rich alicyclic molecules. This is just a proposed structure. It's not a definite structure. Okay, so in order to explore those diversity or cocktail of the organic substances, we're gonna use multiple analytical techniques. And today I'm only focusing on one of them, right? For the ultra high resolution mass spectrometry, the Fourier transform ion cyclotron mass spectrometry, which can help us to explore the DOM at the molecular fingerprint level. So we know the mass spectrometry is to measure M over Z, right? It's like a balance. If the charge is 1, then we are measuring the accurate mass. So for the traditional mass spectrometry, as demonstrated by the green line at the M over Z 380 line, we only get 1 peak, right? We cannot separate things. But if we have ultra high resolution, we can identify dozens of peaks, different molecular formulas, right? So it's like we are taking an ultra high resolution figure for those marine organic matter, right? So with those ultra high resolution, we can identify tens of thousands of molecular formulas, which can help us to better understand the DOM cycling in the ocean. So with those vast of the data, we can present them in the Van equivalent diagram space. And this can help us to simplify those complex organic molecules into some biochemical components, such as something you may be familiar with, such as the lignin, the lipid, the protein, etc. This is really excellent idea to simplify things from a complex, a composition data into some subfractions. However, this kind of classification is operational, and another thing, this classification is not relating the stability of those molecular formula, right? So another colleagues, which I'm gonna show you, is trying to identify the recalcitrant organic molecules from this complex organic pool, and they found the CRAM is an indicator of marine recalcitrant DOM. So actually, this is from a pioneering study from the Professor Robert Hepcorn, and with several other excellent scientists, such as Philip Copeland, the Ron Benner, and John Hedges, who unfortunately passed away in year 2002, and we all miss him. So this paper is a characterization of a major refractory component of marine DOM using the 2 state-of-art technologies, including the FTIC-RMS and NMR. Okay, so based on the complementary advantage of these 2 techniques, they identify a region within this Van equivalent diagram space as the CRAM within this region. Okay, so they provide some potential structure for some of these representative formulas, including they have a alicyclic, right, and carboxyl-rich. So they are short as the CRAM. So this work has been highly cited for about 1,000 times since then. However, recently, some following studies realized that the CRM may not be that refractory as previously believed, as they can be rapidly synthesized by heterotrophic bacteria and some chemoautotrophs. So there's 2 representative studies. The left one is showing you that with 29 days incubation of the bacteria, they can, emit some metabolites which are similar as CRAM, as demonstrated by those 2 panels. And the right panel is from Gerhard Hendel's group. They identify some, compounds synthesized by released by AOA, the ammonia oxidizing archaea. Also here, the archaea DOM is also really similar to the oceanic DOM in the open ocean. So somehow the CRM may not be that refractory, and those are 2 representative studies. And another recent study by the Brad Walker's group, they realized using the proton NMR from the Baffin's Bay, and they realized that CRM is also comprise the labile and refractory molecules. Okay, so our key question is, because this is from NMR, they cannot go into the detailed molecular level just by some, subfractions. We kind of wondering which CRAM are labile and what are their chemical properties. So our hypothesis is that there's lots of, each molecular formula may have multiple isomers, and this isomeric composition may determine its bioavailability or stability, right? Because maybe microbes maybe preferentially utilize some specific isomer, but not the others, okay? So this call our attention. So how we gonna identify the isomeric composition within each molecular formula we obtained from our conventional FTI-CRMS analysis? Because the conventional analysis is based on the direct infusion, which we get lots of molecular formulas, but without any isomeric information. So here we hyphened the liquid chromatography versus the FTI-CRMS, which can help us to obtain the isomeric composition and which also linked with the polarity of each isomers. So this is really in the technical detail. So traditionally for the LC mass spectrometry analysis, you will use the standard gradient, but our colleagues in UFZ, they were using the counter gradient, which can provide us a stable, eluent, that's a methanol, and which can help us to better identify those high polar formulas, or we can expand our detection, window, which can help us to better obtain the more comprehensive molecular formula composition and isomeric composition. Okay, I don't want to go into the detail about the technology part. So to simplify things, right? So we have the LC, then we can obtain a different segment with decreasing polarity. So this is a total ion chromatogram we will get from a oceanic DOM. So looks like we cannot separate things, but that's the real case. It's a super complex organic mixture. So however, we can separate them by 1 millet of LC, which can help us to identify the potential isomeric cluster or feature. So based on our prior knowledge and some standard analysis, okay? So then previously we will only get the 1 mass spectrum, but now if we separate them by the LC as a 1 millet segment, we can get 9, right? So if you check here, previously the data size is only 0.2 gigabyte, which is easy to analyze. But now for one single sample measurement, we will get 25 gigabyte of data. So which really calls attention that you really need a very fast and accurate algorithm to really interpret those data. So here I'm demonstrating using one case to why this is really important. So if you only have the DIFT, so previously, for instance, the formula A in the sample 1 may be the same as formula A in sample 2, right? Because they are same formula. So you cannot differentiate whether they are the same structure or not. But if we have the isomeric information, so if they are present in the first segment, then we assign as 1. So for instance, for this one, it's a vector, like an isomeric vector as 1, 1, 1, and then the other are 0, which means they are not detected. But for the sample 2, the isomer are detected at the later elution time. So which means the top A is not the bottom A. So that's the unique strength of the LC separation and the isomeric composition. So here I'm going to using several cases to demonstrate the strength of this advanced technique. Okay, so here we really want to understand what kind of CRM are labile and what the other are recalcitrant. So we want to use a microbial incubation experiment to help us understand that. So we following the study from Lian et al. At 2021 environmental microbiology, we did 3 incubations. The first one is just coastal seawater, and the second one we amended with the diatom DOM, and the third one is amended with the sediment DOM. So with the microbes, the in situ microbes. Why we want to do that? Because we want to simulate the biological carbon pump with the algal bloom, for instance, right? And this one with the sediment DOM, we simulate like there is anything coming from the resuspension of the sediment with the addition of the DOM source. So then in addition to analyze it by DIFT, we also analyze the sample by LC-FTI-CRMS, and we identify the microbially labeled and microbially produced DOM. So this is a 90 days incubation. So we operationally define any formulas that only present in the beginning is a microbially labeled, but any formulas that only present at the 90 days is microbially produced. Okay, so it's an operational definition. So now, then one important feature is here, we have the 3 groups of incubations, and this is also the total assigned formulas intensity or similar as the TIC. So the dashed line is the in the beginning, and the solid line is after the incubation. So one important feature is for each group, for instance, let's compare the red line, the green line versus the green dashed line. So after incubation, all the polarity increased, right? They show up at the early retention time. So that's a very important finding. We observed that the polarity increase with the microbial reworking or transformation. So then we also compare the LCFT data versus the DIFT data. So we're pulling out the labeled DOM formulas versus the produced DOM formulas. So they look somehow similar. So that further justify the technology as the solid technology of the LCFT. But we got another information, the time dimension or segment information, right? So they have different colors. So some formulas may be present in early retention time, some may be in the later retention time, right? So which can help us to obtain, the isomeric composition. So if we deconvolute the panel A and panel C, it's gonna be like this, right? So previously you only have a formula information, but now they can present in different segments, which can help us to obtain the isomeric information, okay? So you can easily see for the label formulas, they somehow in each segment, they were present in the left top part of the van equivalent diagram, but for the produced formula that were present in the right bottom, mostly right bottom space of the van equivalent diagram. So that's the main characteristics. So then we can also pulling out, by number. So we're pulling out the number of label formulas versus the number of produced formulas in each segment. So still, right, the label formulas showing at the later retention time with low polarity, and the produced formulas showing at the early retention time with high polarity. Not only for the DOM fingerprint, we target on the CRAM. It also shows the same thing, right? The CRAM, the label ones showed at later retention time, and the produced one showing at the early retention time for all 3 incubations with different substrates. So then we can draw all those formulas in the equivalent diagram space, right? The A and B and C are the label CRAM, and D, E, F are the produced CRAM. So you can easily see, right, the label ones in the left top part, and the produced ones, there's several produced ones in the bottom right part, okay? So it's like this way, increasing polarity, okay? So then we further, did some statistic analysis. We're pulling out those formulas and we calculate some parameters, including the aromaticity index, the double bond equivalent, and NOAC, the nominal oxidation state of carbon. So we can easily visualize the produced formulas share the higher aromaticity index, higher DBE, and higher nominal oxidation state of carbon, which further justifies its biological recalcitrance for those produced CRAM. And this is just a one, representative structure proposed by Headcorn et al. At 0.006. So as you can see, they have multiple rings. And for some rings, they may have some double bonds, and they have multiple COOH group, okay, the carboxylic group. So these should have, the further, if we can make a speculation, right, for the produced CRM, AM, maybe they have more double bonds or more rings, okay, in order to get a higher AI and higher double bond equivalence. So then we really wonder, right, if the produced CRM are more recalcitrant in the field. So what we did is just a lab incubation for 90 days, right? We really want to verify if we can validate this finding in the field. So we did a field verification or extrapolation. So we pulling out those labeled CRAM as demonstrated here, and the produced CRM, which are shared by all the three groups of incubations. And one striking phenomenon is that the labeled ones have both oxygen, nitrogen, sulfur, and phosphor, which are reasonable. But for the produced CRM, the recalcitrant ones, they have high nitrogen content, okay? So which suggests the nitrogen, dissolved organic nitrogen, may be an important form of RDON in the ocean. So then we further validate those formulas and detect whether those formulas can be present in the global ocean molecular fingerprint database. So we compiled 1,485 marine DOM samples, and then we try to detect whether those commonly labeled CRM are widely detected in the ocean, or the commonly produced CRM, whether they can be widely detected. So this is just a detection ratio, which means if we have 100 commonly produced CRM and 90% of them can be detected across the global ocean, which can further justify its environmental persistence, right? So then we can calculate the detection ratio for those labeled CRM versus this, recalcitrant CRM, and you can easily visualize, right? The produced CRM show higher detection frequency, which is about 86%, which is significantly higher than the commonly labeled CRM. So then it's a summary for this case. Using this LC-FTI-CRMS, we can really demonstrate a microbial preferentially utilization of low polarity DOM and production of high polarity DOM. And even within CRAM, the chart characterized by the low polarity, lower oxidation state, and a greater abundance of condensed aromatic structure exhibit higher biological lability. And the refractory nature of this microbially reworking CRM provides more direct evidence for the operation of microbial carbon pump in the ocean, which was proposed by Professor Zhang. But we have more direct evidence that they are gradually transforming those low polarity CRM into the high polarity CRM. However, so our study suggests that this calls attention that because there are multiple studies using the CRM as a RDON proxy, not only in the ocean, but also in the freshwater, in the soil. So our study suggests if you only count on the molecular formula information, the CRM may not be a very valid RDON proxy. So then I want to introduce you guys to another two case, why we really care about the polarity or isomeric fingerprint, okay? So let's use another example here, right? So this is also based on the separation and classify into line segments. For instance, as we just mentioned, the formula A in the sample 1 may not be is not the bottom A. So the top A is not the bottom A because they didn't share the same isomeric cluster. Or maybe for the other case, this formula B and in sample A and formula B in sample 2, they share the same isomeric cluster. So the top B is highly likely the bottom B. So that's the importance of isomeric fingerprint, okay? So here, we just want to wonder, right? So for our first case study, we see that the microbes gradually transform those labile CRM into recalcitrant CRM with high polarity. But we don't know if this isomeric feature can really present in the natural seawater. So this is our recent study. For instance, this accumulated positive or accumulated formulas. So with the microbial reworking, the relative abundance increase, okay? So in the day 0, they only share the 5 and 6, those two isomers, okay? This is a vector, okay? You can think about this is a vector. So only 2 isomers were detected in the beginning, but after microbial reworking at 90 days, you can easily visualize there's like 6 isomers. And these 6 isomers are the same in terms of isomeric number at the South China Sea, the top layer. So after microbial reworking, we only matched 33% of the isomeric feature of the natural water for this formula, but after reworking, the matching ratio is 100%. Okay, that's a very important finding. And there's some other formulas which are not present in our incubation before, but after 90 days, the microbes may produce or transform into some new formulas. So previously at day 0, they are not present. But after 90 days, we detect those formula and the isomeric composition is also really close to the natural seawater, which means just by 90 days, the microbes are doing crazy job to synthesize some formulas with the isomeric feature that is exactly or really close to the natural seawater. So we not only compare the surface ocean, we also explore the matching ratio of the deep ocean. It's very similar. So there's a new isomeric evidence suggests the microbial carbon pump is highly efficient. Okay, so this is the first case why we care about the polarity or isomeric feature. So another case is, so previously we were already talking about the qualitative level, but we really want to quantify the source of organic substances, including the source of the oceanic RDOM, right? Because that's also a mystery, because there's lots of speculation that the dominant contribution should be, the biological carbon pump that provides the initial source. Then with the microbial carbon pump, they transform those organic substances into the RDOM, right? But it's really hard to do the quantification. So here, we're using the molecular isomeric information. We can help us to do some quantification, okay? So here, we really want to quantify if riverine input is a significant source to the oceanic RDOM. Why that is important? Because there's lots of Earth marine carbon cycling model. So usually they will assume the riverine input is a minor component contributing to the oceanic refractory dissolved organic carbon pool, right? But there is no direct evidence. There is no solid evidence to prove that. So here, by using this, polarity or isomeric fingerprints, we can really pulling out the recalcitrant terrestrial molecular fingerprint in the ocean. And based on this set of molecular fingerprint of those formulas, and based on some bulk measurement, including the dissolved organic carbon concentration and radiocarbon isotope measurement, we can semi-quantitatively evaluate the recalcitrant dissolved organic carbon that is discharged by the river and eventually contribute to the oceanic RDOM. So based on this study, based on our rough calculation, we estimate that within the oceanic RDOC pool, there is 17 to 25% within this pool is highly likely contributed by the river input. Okay, so which highlighting the lead to adjust the global carbon cycling models. So the take-home messages of these 3 case studies. The first one, so compare with the molecular level, formula level alone, if we add in the isomeric or polarity dimensions, can significantly improve our understanding of the biogeochemical cycling of DOM, right? And these dimensions can help us pulling out the interested or scientifically significant formulas. And why we need that? Because we really want to, evaluate, better constrain the source, better constrain the biogeochemical cycling processes, the underlying mechanisms, etc., right? But there's still several limitations, not only limited to the following here. So number one, what we are talking now is just the chemistry part, right? We have limited biological evidence, and especially at the genomics level and transcriptomics level. So I'm really happy to do more collaborations with any one of you if you are interested in this part. And another thing is we are still talking about isomer clusters. So we don't have a definite structure diversity of the marine DOM or RDOM. So this is our ongoing dreams, okay? So with that, so there is a call for the papers. We organize a special session about the carbon cycling from the land to ocean. And with that, I want to thank you for your attention, and I'm more than happy to take any questions.
All right. Thank you so much, Ding, for an awesome presentation. And thank you for sharing with us your dream. We're here to take part in dreaming.
Everyone is a dreamer.
Right. Yes. Okay. Now, let's move on to our panel discussion. Again, joining us are Professor Jiying Li from HKUST and Professor Penghui Li from Sailybayev University. And of course, Ding, you stay with us for our panel discussion. All right. So Professor Penghui Li, we'll call you Penghui from now. I'll begin with you because you share very similar research interest with Professor Ding He in characterizing the molecular structure of dissolved organic matter, particularly the recalcitrant DOC, RDOC. So we have also done research on your work. You recently published a paper in Science of the Total Environment. That paper provides really valuable insights using direct infusion, Fourier transform, ion cyclotron resonance mass spectrometry. We call it DIFT ICRMS. In contrast, Professor He's work in his Science Advances employs online counter-gradient liquid chromatograph coupled to FT-RCMS, which adds really a good dimension of separating DOM based on polarity prior to spectroscopy. Can you comment on the key differences between these two methods? That'll be the first question. In other words, can DI-FTRCMS acquire a similar, structure versus the LC FTRCMS? Penghui? Here?
Recording in
formula.
progress.
So just by the the possibility for each formula, for some formula, they may have several thousand or even more structural possibility, right? So that's
Right.
still another limitation for this current technology. But we do use the mass, okay? So the triple, quadruple, which can help us to get more accurate structural information.
Okay. All right. We welcome Penghui back. Penghui, can you hear us?
Sorry for the interruption. Yeah.
Okay. All right. We talked about the two methods. One is direct infusion. The other one is LC component. Can you, because you have a lot of experience in doing FTRCMS, can you please comment on the disadvantages or advantages of each method? Any,
Yes. Thank you for Professor Zhang for the invitation and also for the great talk by Ding He. For the question, I think compared to the DFT, as LCFT can give us detailed information about maybe because in incorporating the return time, providing the polarity, which could enhance our understanding of the DRAM composition and thus the bioavailability. But only few labs have this instrument. So for most researchers, we don't have access to this instrument. So maybe in the foreseeable future, the DFT will play a dominant role in characterization of DRAM and elucidating of the DRAM biogeochemical. But from Ding's talk, we can combine the DFT and LCFT. Maybe we can integrate machine learning and other techniques to infer the polarity related information from the DFT. And even the DFT, which could not provide the polarity information, but which can give us the detailed molecule composition. For our paper, we demonstrate the carbon oxidation state and also aromaticity dominant the bioavailability of DOM, which is consistent with the papers by the Dr. Tsai and Professor He. I think maybe sometimes we can combine these two, but nowadays we don't have this access. But I think under this scenario, the DFT could play a role in the current time.
Right. Maybe you can expand on the polarity of DOM. Is polarity the currently most effective way to delineate the CRAMs?
Yes.
You can join the discussion because the polarity seems to be a very effective way to give you a good picture of different isomeric features of DOM.
Yes. I know the polarity or the isomeric diversity can increase the DOM diversity, so that the dilution hypothesis may play a role. But for the polarity, from test paper, we can know the polarity play a role. But what's the mechanism under this scenario? We know polarity is only a characteristic of the DOM, which may be determined by its molecular structure.
Right.
So the bioavailability is determined by the polarity or finally by the DOM structure. So
I think
we will know the DRM the polarity is mostly caused by the carboxyl or hydroxyl. You know, the carboxyl also increase the carbon oxidation. The carbon oxidation state, which also hinders the utilization of DRM. So which is the fellow reason for the DRM recalcitrance? We don't know. Maybe sometimes we need more details about the DRM structure. Yeah.
Right.
I think yeah. We will need to combine.
You touched upon a very good point about the mechanism of a compound being recalcitrant. Is it the polarity or is it the structure? Can you comment on that?
Different structure has
polarity.
different polarity. So we cannot, solve the accurate structure at this point. So we can only represent it by the polarity currently. But as I said, so in the future, because currently the LC-FTI CRMS is really expensive, about $3 million, right? Or even more than that. But you really need experienced people to do the measurement, do the method development. But in the future, I really see, right, the high potential of Orbitrap mass spectrometry and the LC Orbitrap mass spectrometry is, let's say, only less than $1 billion or even less. Okay, so which are widely used for lots of labs, especially in the biomedical field, right? They identify the peptides, right? Those. So I do see we can have better structural information in the near future, and which can help us to better understand some target of interest formulas.
Okay, excellent. We look forward to new technologies to come online to help us with the identification of structure versus polarity. Well, thank you so much for those insightful points, both Penghui and Jiying. Now let's turn to Professor Jiying Li. While Penghui focuses on water column DOM, we know you have been pioneering research exploring biogeochemical processes at the sediment-water interface, particularly cycling of phosphorus that may influence by organic matter composition. A fundamental question for you may be whether sediment can act as a source or sink of DOM, particularly in coastal systems.
So whether sediment is a source or sink, so it depends on how you think about it. So if we are looking at specifically DOM in the water column and we want to understand what controls the concentration in the water column or the composition. So in this perspective, sediments are sources. Well, because they receive the organic matter from the water column, right? And then associate with the degradation of that accumulated particular organic matter, then DOM is produced. And some DOM will be released back into the water column. So from that perspective, it's a source of DOM. But if we are thinking about maybe the carbon budget or the cycling of organic matter as a whole or in general, sediments are net sinks of organic matter in general. Not just because they buried or decomposed the particular organic matter, but they also, some DOM can be sought from some sediment matrix or incorporated into sediment minerals and can be permanently buried or into the long-term geological cycle. So from these depending on how you think about it. So from this perspective, well, you can think of it as both sources and sinks, depending on what questions you are asking.
Right. I like to really present a challenge to you guys by asking a question related to the paleo ocean, DOM. We have to study the sediment record, and not now by studying just the extent water column. Is there a way for us to elucidate what was going on 100 million years ago in the water column by studying a sedimentary record?
That's an excellent question. So actually, Professor He and I were talking about this a bit. So a fundamental challenge for using sediments as an archive is that diagenesis happens. Diagenesis means when the material reach the sediment, it's not gonna stay there forever. It's changing. So you may measure the composition of the DOM and you date the sediment to maybe it's a very long record, but then it's difficult to tease apart the historical record and the change after the diagenesis. So this is the major challenge. So a good way is to look for those conservative molecules. So if we know, maybe by doing some incubation or some other way, if we know that some DOM are somehow protected, maybe by some sediment mineral matrix or somehow, so they can be relatively well preserved in the sediment, then we can use those as a proxy to understand those DOM in the water column in the past.
Okay.
It's very challenging.
Yeah. Well, I hope with challenges ahead, there will be innovative thinking, innovative technologies to address those questions. All right. Now we will, let's see, end this panel discussion for now, but let's see if we have any questions from the audience. I know Yernar has already had a wonderful question regarding the Caspian Sea. I know Caspian Sea is your really favorite place to study anything. You want to pose your question again to people here?
Yes. Thank you, Professor Chuanlun Zhang, for giving me the privilege to co-organize this seminar series. I represent the Caspian Environmental Consortium, so I'm using every chance to ask our speakers a question from the perspective of the Caspian Sea. I will explain. Given the Caspian Sea is a fragile closed basin experiencing significant changes in level and salinity, how can we can the advanced molecular technologies discussed today help us monitor the health and predict the future carbon cycling of this ecosystem under climate change and human pressure? Thank you.
Jiying, you want to take on that question?
It's very challenging because, as I said, right, because this is a micro scale study. So we want to understand some underlying mechanisms. But if you really want to project what's going to happen in the future, you're going to need lots of data, right? So you're going to do lots of observations and try to link those observations with some, bulk parameter. For instance, future CO2 concentration, future temperature, or future some bulk parameters. So, and then you also really want to get a representative samples. So from our, mechanistic study, So we want to pulling out what's the dominant mechanisms. So then you want to consider this kind of mechanisms in your future experimental setup. But our last case study is doing some quantification, but there's not still lots of limitation, right? So so we want to better refine our current, source of quantification, and then we have better understanding what's happening now. And maybe with the, the sediment record, we have a historical evolution, and then with those kind of data in hand, maybe with the AI technology, we can make prediction what's going to happen in the future. So, but I mean, it's a very broad answer, but it's very challenging.
Yes, thank you very much. Thank you, Dika.
All right, so let's see. It looks like we have a question from the chat room. Ding, can you read the message in the chat room?
How did you match the DIFT data with the LCFT data? Was that through linking the molecular formula detected in both data or other way? So yeah, so actually for most of our case, we will detect more unique formulas in our LCFT ICR measurement for the same sample. The reason is, we can somehow decrease the matrix effect with the LC separation. So then we can increase our analytical window. So then after we assign the molecular formula, then we can link those two techniques. Okay, so that's the first one. The second one, as you have shown in this paper, some CRMs also have relatively low OC ratio, which could be indicative of relative label. How do you interpret these interesting results? I think there's still lots of mysteries, but my rough, answer is the transformation, since we have the oxygen. So the dominant process will be gradual oxidation. So we gradually adding the oxygen to your molecule. But to some point, if you add too much, then you're gonna, transform that, degraded them into the CO2, right? That's the highest oxidative state for the carbon. So, but for below a certain criteria or limit, gradual oxidation may increase its biological recalcitrance. So that's my answer.
Well, this actually brings up another interesting question about the oxidation state of DOM. I know the best oxidative environment will be the water column. In the sediments, under anaerobic conditions, the oxidation is expected to be very slow. Whether it's slow or fast, does it impact the ultimate oxidation state? Whether the oxygen is from O2 or from nitrate or from sulfate, doesn't matter. Was it where oxygen comes from?
Yeah, that's a very good question. So that's the thing we are exploring with Professor Li. There's lots of oxidant, right? Not only oxygen, but also maybe sulfate, etc. So maybe there's some other, mechanisms in the sediments.
Yeah. Okay. Well, there are so many interesting questions, particularly those related to microbial processes we have not touched on. I do like the audience with a molecular or microbiology background to share a couple of questions from my students who are interested in the microbial aspects of the DOM degradation. I wonder if, Ding, you can quit your sharing of screen so I can share a couple of slides.
Okay, sure. Hold on. Yeah, here we go.
All right. Let's see what I have here. Okay, this is one of those questions related to microbial processes. Is the transformation achieved by microorganisms modifying existing CRAM framework? or do microorganisms utilize degradation products to synthesize new, structurally more intricate CRAM molecules de novo?
Okay, so actually this is a very excellent question from the, I guess the UG student, right? Undergraduate, right? So it's a very good question. And it's a very tough one. So every techniques has limitations. So in our case, in the initial incubation, the seawater only contains a small fraction of labile CRAM compounds, right? Because they disappear after the 90 days incubation. We believe they were not completely transformed into the inorganic carbon or inorganic matter. Maybe they were modified into the more recalcitrant CRAM, which are characterized by our different parameters, including the higher oxidation state or highly aromatic structure. So as we all know, any single labile organic substrate can produce diverse CRAM compounds. If you can look the Leckenfeld et al. 2015 Nature Communication paper or Professor Zhang's National Science Review paper in 2018, we believe microorganisms were utilizing various organic substrates to produce the recalcitrant CRAM. So both scenarios will exist, I guess.
Great. Yeah. Well, we call on microbiologists to really join Ding's dream team to really study the fate and polarity of DOM from both the chemical structure and also the mechanisms underlying its degradation. So I think our time is up with this session. I hope we can all take home message by today's presentation to either give it a good shot at the tough structure question or the microbial processes utilizing either transformation of existing CRAMs or starting from scratch to build new CRAM molecules. Those are really open, open questions for anyone to think about and also work on it. All right, with all this wonderful presentation, discussion, and sharing by the audience, we thank the speakers, the panelists, and we thank the audience for joining today's seminar. Okay, thank you so much. Goodbye, everybody.
Thank you. Thank you, everyone. Thank you. Bye.
Bye.